right? So if you are smelling cannabis and you smell coffee, there's not just like one terpene that is making that cannabis smell, or you detect sort of a coffee scent. It's a combination of. This viroid is highly infectious. It can be transmitted through clones and seeds from infected plants. So it's worth looking into these beneficial bugs in order to prevent the invasion or the infestation of other bugs and pathogens.
So usually they think indoor high quality, outdoor low quality. But again, really, is this really true? And the genotype contributes to the phenotype. Contributes, okay? And we're gonna talk about that. It is not the phenotype, it contributes to the phenotype. We want plants that are very different from each other, that are very diverse, that have different colors, right? So here, this plant has these yellow pistils, this one has pink, this one has white, but you can
we do know that Cannabis sativa and Humulus lupulus, which is hops, both came from the common ancestor. This is the reproductive structure, and upon fertilization, or upon pollination, this is what gets fertilized and will then become a seed.
And it produces exact copies of the plant, right? Sometimes you can produce plants without disease, for example, viruses or viroids. I don't know if you've heard about HLB.
So climate control, which is one of the most attractive things about indoor growing, is that you get to control the environment.
What is a genetic map? A genetic map is when we take that assembly and we associate particular locations of that assembly to a trait.
But a primer is just a little fragment of DNA that's going to stick to the single strand of DNA and then initiate the polymerase to start reading and adding nucleotides.
And that is crucial for consistency. If you want consistent plants, if you want all of your plants to be the same color, the same height, the same, etc.
So you could actually do like a PCR test to come up with what kind of microbes are in the sample, because microbes have DNA. And if you test positive with microbial DNA,
we have here alpha pinene and then here beta pinene, and you can see here the double bond, right? So they're basically the same chemical formula, however the double bond differs.